Hello, my name is Alia Al-Assaf. I'm an evidence synthesis specialist at the NIHR Innovation Observatory. This presentation is about searching preprint repositories for COVID-19 therapeutics using a semi-automated text mining tool. Please take some time to read through these acknowledgements and disclosures before I move on to the next slide. Introduction. The National Institute for Health Research Innovation Observatory, NIHR Innovation Observatory, is publicly funded by the National Institute for Health Research, NIHR, which is the research arm of the National Health Service, NHS. The NIHR Innovation Observatory is a horizon scanning, research and intelligence center based at Newcastle University, with an academic home in the Population Health Sciences Institute. As part of our remit, we deliver a national horizon scanning and early awareness notification service to national key stakeholders and decision makers, among which National Institute for Health and Care Excellence, NICE, NHS England and NHS Improvement, and NHS Accelerated Access Collaborative. Searching preprint repositories for COVID-19 therapeutics. Since March 2020, the NIHR Innovation Observatory has been providing primary horizon scanning intelligence on COVID-19 therapeutics for key stakeholders in the UK, and those include Research to Access Pathway for Investigational Drugs for COVID-19, Rapid C19, Department of Health and Social Care Therapeutics Task Force, UK COVID-19 Therapeutics Advisory Panel, and National Prophylaxis Oversight Group. The NIHR Innovation Observatory website provides a dashboard of COVID-19 therapeutic interventions in clinical trials for treatment and/or prevention of COVID-19. The information on this dashboard is updated regularly on a weekly basis. This dashboard can be accessed via the following link. Early detection of positive or negative signals from preprint sources was a crucial part of the intelligence provided to alert the system of potential COVID-19 therapeutics. Preprint sources were crucial due to the very noisy and rapidly changing landscape to address the urgent public health situation caused by the pandemic. So we developed a semi-automated text mining tool in Python 3 programming language. The tool uses the IDs of ongoing and completed clinical trials that were already selected from major clinical trial registries to search to preprint repositories. These repositories are medRxiv and bioRxiv. We decided to search those two repositories because of the language, advanced searching criteria, and because of the featured topics in these two repositories. This diagram shows the process that we have used to provide our stakeholders with intelligence on COVID-19 therapeutics. The NIHR Innovation Observatory scans for clinical trials through searching major trial registries, and these are clinicaltrials.gov, European Clinical Trials Register, and the WHO International Clinical Trials Registry platform. Only studies with interventional design would be included. Studies with other design would be excluded. Then only trials that have an intervention on treatment and/or prevention of COVID-19 would be included. Other types of interventions, such as diagnostics, for example, will not be included. From these included trials, every week the tool that we have developed automatically collects the IDs of clinical trials that have ongoing statuses, such as recruiting, active not recruiting, ongoing, etc., or trials with completed statuses, and will exclude IDs of clinical trials with other statuses. Then the tool will use the primary ID of these included trials to search medRxiv and bioRxiv to find preprints with matching trial IDs. It will repeat the process using other IDs of the clinical trials, in other words, secondary IDs, if no matching preprints were found when searching with the primary ID of the trial. The tool will exclude preprints on reviews, such as overviews on ongoing clinical trials. It will extract information such as title, authors, publication date, URL, and DOI from the selected preprints, and will produce an output. This output needs to be manually screened. Only preprints that report results from clinical trials or other types of studies, such as observational studies, for example, on COVID-19 therapeutics, will be included. Why searching preprints rather than searching data sources for journal articles? To identify clinical trial results faster, to help stakeholders detect potential therapeutics that may progress more quickly, ultimately speeding up patients' access. The timeline for publishing a journal article may take several months. Also, the timelines between disseminating results in preprints and publishing results in a journal article could take few months. Therefore, searching preprints can help identifying results as soon as they are disseminated. Findings. We found results disseminated in preprints for 155 out of 3,304 of the selected clinical trials. We've included 177 preprints reporting clinical trials results, and 94 interventions were reported. Some of these interventions are being investigated by multiple trials. This chart shows the 5 most frequently reported COVID-19 therapeutic interventions from the included preprints of clinical trials results. The most frequently reported intervention is convalescent plasma, which makes 40%, followed by hydroxychloroquine, which makes 30% of the 5 most frequently reported therapeutic interventions. We've also included 118 preprints that presented results of other study types on COVID-19 therapeutics. From these studies, 70 therapeutics were reported, some of which are being explored by more than one study. This chart shows the 5 most frequently reported COVID-19 therapeutics, that is, medicinal products including vaccines, from the included results of studies other than clinical trials. The most frequently reported therapeutic is tocilizumab, which makes 1/3 of the 5 most frequently reported therapeutics, followed by hydroxychloroquine, which makes 26% of the 5 most frequently reported therapeutics. There is a limitation in the method that we use to identify the preprints, which is the possibility of missing results of some clinical trials. For example, results that could be disseminated in other sources, such as other preprint repositories. Conclusions. Using text mining for identification of clinical trial results proved to be an efficient approach to deal with great volume of information. We've also concluded that human input is essential to validate the tool outputs. Thank you.